Well, if we were doing this together in class, you'd all have your own gummy worms as chromosomes. Just to point out, each gummy worm, right, has two color segments. Those represent their genes. So, during interphase of meiosis, now we're making sept cells, all of the DNA or chromosomes double. Boom! So now we have twice as much. And prophase starts by these pairing up and making homologous pairs. And they're going to be absolutely identical, aligned gene by gene. When these pairs find each other, we call this synapsis. They pair or they hook. It's a loose hook, but they are sort of magnetically stuck together. So these are homologous, the same chromosomes. As we proceed in prophase 1, remember prophase 1 is 90% of meiosis. It's very long because a lot happens. These homologous pairs find another pair and they form a set of 4, and we call this a tetrad. So now we have 4 chromosomes hanging out together. Then, as prophase 1 proceeds, this amazing process that allows for a ton of diversity in populations, crossing over happens. And so to do this, to show you this, I'm going to move these two out of the way. This wouldn't happen, they would stay there. And these literally just cross over. So one end crosses over the other. This structure, this crossed over structure is called a chiasmata. And this could happen at multiple places. So it could be right, it could be happening here, it could be happening here, it can happen multiple places. There could be 4 or 5 that happen. And so what really occurs is they're going to exchange part of their DNA. And so now, this is why I love gummy worms, because they're going to stick together just like our DNA would. Now, we still have a tetrad, but we no longer have homologous pairs because crossing over occurred. And once this has happened, we have this new diverse DNA. We have some of them that are original, but we have new recombinant, right, recombined DNA chromosomes. This is why in a family, sometimes you look a lot like one sibling and hardly anything like another, because this diversifies a parent's DNA. So this is crossing over, and this happens during prophase 1 of meiosis.